Since we have a few people, like we should call Rahul also.
He wanted to come, but he's giving a talk at Ashoka today, so he's not in town.
Yeah, because our Ted is also not there actually. He has gone for he's out of Delhi actually. So Dr. Card. We can just see that. We can wait. Just wait for 5 minutes.
Yeah, no problem.
Pleasure to invite Professor Vinod Kumar from TU University. And it was a surprise to me, like a couple of weeks back, almost 2-3 weeks back, I got an email from him saying that he has read my paper in class 1 about circadian biology, and he would like me to come to his lab to have a discussion about circadian biology. Then I said that I asked him basically whether it's possible for him to come here basically to give you a talk, and he obliged basically, and so he's here. And most importantly, he's a recipient of many awards. Already it is there in the flyer that which we have sent you to. So I don't want to read basically because the flyer is already given to all the people. And to just name a few that he has actually he got several international fellowships under INSA, that is the Indian National Science Academy. And one of the very important award is that he was a 2000 Young Investigator. He got a 2000 Young Investigator Award Gordon Research Conference in Pineal Cell Biology. Okay, so and also he was recognized in various international forums. And most importantly, that his interest is basically in circadian biology, and he is also the president for the Indian Society for Chronobiology. Okay, without much ado, I'll call upon Professor Vinod Kumar to just give his talk. Hopefully, the students come and join.
No problem. Thank you, Dr. Sriram. So when he wrote me to come, I wrote him back saying that it's a computational biology and engineering, and except my son is here, I have no person with the engineering, and he also never tells me what he does because he thinks that. But nevertheless, he said that I should come. So talk is not very organized. I will be, a part very general because I thought, and then part is specifics. And for my own interest, I work only on birds, although I've worked on mammals, large mammal and small mammal, and sheep, rat, hamster, and everybody. But finally, since my PhD days for 40 years, which this year I completed, for 40 years I only work on birds, and I have my own reason why I work on birds. Now, nowadays, for the last 20 years, I don't think I work. I only write grants and papers, and these people work. And therefore, they not only what's very important that they, teach me also. I mean, otherwise we all know your teachers that you would not teach. I would not learn it unless they want me to learn. Peers, which is very important because if you don't get the criticism, you never improve. And then senior colleagues and mentors are always there in life. The lab works several things. So I'm a university professor, and I always argue in our own setup, in Indian setup, that there are two types. You do research, either you are very brainy and go vertical and produce a product. And if you cannot do it, if you are a university professor and limited with resources as we are, so you do the horizontal, you train a generation of students and who can really work. So at least at the same time, 5 different streams work in the lab, and so we also work on behavior and life history, how the animal organizes. Recently, a paper came in Animal Behavior, which is the journal of the animal behavior. We also work on the seasonal reproduction and migration, endocrinology, neuroendocrinology. Regularly we publish in these journals. We also work on the brain, so we work on neurotransmitters, neurogenesis, and so on and so forth. Then I've never been a molecular biologist, but recently we have been working on molecular biology very heavily, and also cognition with a specific model system. Now, the central question, which has been very interesting, and really when I joined my PhD 40 years ago, chronobiology was not in the picture until 1979 when Dr. M.K. Chandrasekhar got Bhatnagar Award. So he was invited in BHU. I was a student, so I was very much fascinated without understanding that something is here. And then in '84, when I got the job, then I went all the way sleeper class to Madurai to learn from him. And it took a long time, but then this question was always in my mind, that how the organisms achieve the precision in activity. So you can do the activities, but the precision is very important, and precision, and we all evolutionary biologists know that if you do there is always luck factor or chance factor. So the right time is very important. The question is very important, that how would one know the right time? So how then the right time is known? Because if you are not an astrologist, you can't even predict unless you are a computational biologist. So you will predict. So therefore, how the right time is known? But the problem is, what is time? I mean, I am not sure that, and this time I'm reminded, and these mythology, our mythologies are beautifully placed with many quotations, and they are very helpful at the time to understand. So I vividly remember in Ramayana, in some, so Ravana was changing the kal, time. So he could get every, devata in his prison, but he was unable to imprison the time, because his concept was very simple. If I don't let the time move, then I'm immortal. Doesn't matter, I wouldn't age. But he could never. And if you see our depiction, the time has 7 chariot, 7 horses chariot. No other has. So therefore, this is very, so we don't know the time. Honestly, nobody knows. And because if nobody asks you or me, then everybody thinks that I know the time. The problem is, if you want me to explain, then I will have only one answer, that I don't know. And to me, nobody knows what is time anyway, except if you are a poet. Because if you are a thinker, poet, a spiritual person, you have, so between night and day, and an explore territory, not shadow, not light, it is time. So when we have nothing, we have no knowledge, we build up the hypothesis, right? So therefore we need to have some hypothesis. So we can divide into several, say, okay, let's say, so whatever we do about the time, we do time as a sense, time as learning, time as a scale, time as a place in the brain, time as culture, time as cycle, time as draw. I mean, you can go on and put this, prefix time, and you define, because with everything the time concept would change. Now, let's see some of the, just a couple of examples here. Let's see time as a learning. So this is an internet downloaded picture, and the 3 people are waiting, in a dentist room, let's say, or for anything, or for their Baba say or whatever. And you see all the 3. Do you find any difference in all the 3 people? Their facial expression is different. They are all waiting. They are waiting in a different way, because we know that this is what happens. Everybody waits for the time, but the way you perceive time is very different. So learning, fear, happiness, neutral, disgust, sadness, shame, everything. So the time can change its definition, and I would quote Dr. Einstein here to say this, because I didn't, this is not me, this is him, published 1938, and you can read yourself the abstract, 3 lines of abstracts. So now, what is time? I mean, we don't know what is time. Everybody thinks what is time, but we don't know time. So we try to work one way or the other, because time can have multiple understanding and definition. Let's say time as a scale. That's what we chronobiologists are basically interested. And this also can differ between animals and between the context in which we are talking. So if you say sound tracking or localization, you see, in microsecond, I mean, we all know, if I am standing and somebody speaks from this side or this side, I know whether it is coming from the left or it is coming from right. When you see what is the distance between the 2 cochlea And then what is difference between the auditory cortex anyway? So very small difference. So we have that procedure. We can, you know which direction it is coming, and so therefore it's microsecond. And maybe you compute engineers, you can also go nanoseconds or whatever. This is for just understanding milliseconds. So there is a time in millisecond. Timing is speech generation, recognition, motor control, music perception, working memory, and so on and so forth. But then we have the other time, like interval foraging, conscious time estimation, decision making. So we all are intelligent when we come to a certain level, but then it's our reaction time differs. And that's how we say he's genius, because his reaction time is better. What really is the last one is the circadian time. So like sleep, wake cycle, body temperature, physiological and behavioral function. So by this cartoon diagram, what I'm trying to emphasize, that the time can have multiple magnitude the way it operates in the nature. And honestly, it is everywhere. So it's not in one class of organism, or it's not in one species. So it's everywhere. It's everywhere it is happening if it is a biological organism. But for now, for today, I'm just going to really focus on the circadian timing, and that too in the physiological and behavioral functions, because that's what our lab interest is. And the way I look at that, this is going to be circadian clock, because that measures time, and that means it is going to be about 24-hour timer. And there are two things I look when I look for a clock in biology. It has to be robust. What does that mean? That it should be relatively insensitive to internal changes. It's not that today I'm having fever, so I started measuring the day longer or shorter, or if I go to Himalayas, then my time runs faster. So it cannot be. So, and then also, but if I go to San Francisco from here, which is 13 hours a difference time, then I should be able to adapt to external environment. So, then I also look for that whether it is self-sustained, it can be entrained by an external cue, and it does give the stable output. So these two things we look for when we look in biology, if the biology is being timed and if it is timed in a circadian manner. Now, as the definition would say, that means it is a cellular property, each cells have it. It is also used in the molecular loops, which is feedback loops. Everything comes at the same point where it started, and therefore it says that they don't need cell-to-cell interaction. So it's a cellular property. So this gives really all the concepts which we have nowadays, that you can generate a signal, but if you want a bigger and a smaller signal, you connect it. But this is all in circadian biology. So very simplistic diagram. So each cell have the nucleus and the cytoplasm. There has to be transcription within the nucleus. mRNA messenger RNA has to come into the cytosol. It has to translate into protein, and then the protein from one to another, they can go to the phosphorylation and dephosphorylation, and the time taken, it makes the clock. So this is a simplistic way of putting it. So therefore, to begin with, all circadian clocks are multicellular structure. If you translate into engineering, it is multimolar. Why? Because if there are few moles, their standard deviation will be quite large, because each mole would do its own function. If there are large molecules, then there would be relatively less standard deviation, and the net result will be close to 24 hours. So if you go to the circadian, suprachiasmatic nucleus, each cell has its own clock, but then together they produce it near 24-hour clock. So that's the philosophy the biology has. Now coming to our own, how birds keep their life on time, because for us they are birds, but they are live. They do all sort of things which we do or we can think of doing. So they have the clocks, they show it as output, we call them rhythms, and then they also show it at a phenotypic level that we call as behavior. So the lab tracks all the three. So we track the clocks, we also track the rhythm, and we see how the behavior is getting into structure. So for today, that's the way I titled the talk, clocks, rhythms, and calendars. So how the clocks are there, where are the clocks, if they are shown into a rhythm, and if that clock and rhythm can be used as a calendar, like today is the day and tomorrow is another day. So, but the bird needs to know it. We all need to know. I mean, midnight and midday everywhere throughout the universe the same. It changes at 12 o'clock night. Make no mistakes. The time is the same. We all use it, and we've all formed that calendar. We don't think it because we don't think anyway that what is being changed. This is a cartoon diagram from the book saying that in our brain there is a 2 nuclei which is very small, less than 1 millimeter cube. Each contain about 10,000 cells, so we have 20,000 cells sitting in the brain, very small, in very small area. Each has a clock, each clock ticks and tells the time, but together they produce a 24-hour time, and this time gets synchronized. Here are depicted by the light-dark cycle. So each morning you set up the clock, and that's where you wake up and get up. So there are two phenomena each day we face. In the morning we wake up and get up. It's just, if people are not very lazy, wake up and get up time is the same. If the people are very lazy, then wake up and get up time. It's all ecological significance, because if everybody wakes up and gets up at the same time, think about what is going to have the consequence in the big family, in a house. And I just tell in a public lectures where they are not scientists, many scientists, I have to give some example, and I say even if there is 6 rooms or 6 bed flat, in former times all 6 rooms never had the toilets. Only 2 toilets used to be there, and they were all sufficient because we all have our own rhythm. So somebody needs to go immediately, somebody goes, we all have waking time, we all have sleeping time, we all have every time. So the same way, birds are more complex than what we think. So that is why they conquer almost all the ecosystem of the nature. So they are terrestrial, they are aquatic, they are aerial. The same birds can have the 3 modes if they want. I don't see any mammal can do that much except a few, but almost all birds do the same thing. So they also have the 3 central clocks. One sits in the retina, the other one is in the pineal, and the third one is in the brain. And and they all interact together. They all have the molecules here also in the periphery, almost all organs, and they all give the output, and this has a feedback. And they all get the information from the nature. It's time of the day, time of the feeding, temperature, so on. Social factors. So nowadays, social chronobiology is getting very important. Mondays are always worse than the Fridays, because nobody wants to come at the time. So morning class 9 o'clock, Monday is very bad. So these 3 clocks are there, and this is the rhythm I was talking. So if you
One small clarification in the earlier slide is like the pineal contains basically the ultradian rhythms or circadian?
Yeah, circadian rhythms. Circadian.
So it's basically cortisol and all those things?
No, not cortisol, melatonin. Yeah. And melatonin and cortisol are interrelated.
Yeah, because these are negatively regulating
each other. Melatonin is a very interesting molecule in the sense that nocturnal and diurnal animal have the same profile. And if you see the humans, which we are diurnal, and the rat, they are nocturnals. They are active at night, we are active during the day. I'm not talking of those who are active at night anyway, but that's how the species difference is. So, but both have the melatonin high at night. So it's just a very interesting molecule.
And second question basically is that about the neuroanatomy, because we know that the Drosophila and the mammals, they do have a different anatomy. Do the songbirds or this one do have a different anatomy, and how many number of neurons do they have?
So So clear answer is that nobody has counted in the avian brain, but because avian SCN is a lot different from the, and that's why until now we used to call that avian SCN, because it does not have any retinoid recipient capacity, so it's not connected with the eyes, and therefore there are receptors in the brain itself which give the light, but estimation is that it has 16 to 20,000 neurons. Similar to, yeah. So that's estimation, but nobody has done it. So now, just very interesting, if you observe it, this is a migratory bird. So it shows activity during the daytime, and when in migration it shows night time, but if you see here, it doesn't show anything. So it knows when to stop the activity at night. So even 2 hours before the morning, regularly it stops. And if you remove the day and night from here, you say, okay, I don't put it, that's experiment, and they say this bird is nocturnal. Now, a single species becomes from diurnal to nocturnal to diurnal to nocturnal to diurnal to nocturnal. Now this can be a good non-model system, and that is why DBT funds for it. Otherwise they don't fund any bird work. This is the single project they funded after a lot of hue and cry, because I said it's a non-model system, and here is and some people are good scientists in the committee they were. Anyway, so let's look at the pineal, and this is the cartoon showing that the pineal itself is a clock, and I'll show you a little while why, and it has a rhythmic output. So it always shows the melatonin at night, and in a human case it comes about 3 hours, 3.5 hours after the sunset. That is why we used to have the 9 o'clock news. So 8:45 used to be Hindi news, All India Radio, and 9 o'clock used to be English news, and after English news, no British living here never used to remain wake up. That's the time they used to go to bed, because that's the time when melatonin comes up. So if anybody sleeps at 9:30, even nowadays, it's good, but now we don't have real night anyway. So therefore we can have our own night the way we want, and we can take up later on if somebody is interested. So how the avian pineal cell is a clock, and we published in BMC Genomics, where we made the culture of the pineal cells, confluent culture, and measure the melatonin. So whether this is with day and night or without day and night, and this keeps on showing the rhythmic output. So that's was one major and one testimony saying that the pineal does contain the clock. It is a calendar, because if you change the night length of the night, you change the duration of melatonin secretion. And that's probably the animal knows. Animal measures the length of the night and then says, okay, this is the summer or this is the winter. And they use it, so they use it for their own physiology. So this is a PNS paper we published in long time ago, saying that if you measure in the winter and if you measure in the summer. So if you see the both, there is a significant change in the melatonin profile, both in the duration and the amplitude and the way, because it's not the total production which changes, it's only the distribution of hormone which changes. Now it's not only the blood. If so this is again, you can do the experiment in captivity, and this says that both the things, if you change the intensity of day, you also create the change in the profile. So the point is that the melatonin itself can serve as a calendar. So this is one example to show you that birds not only produce the melatonin, not the melatonin rhythm, so they have pineal clock, they have the rhythm, and they use it as a calendar.
So there is one clarification.
Yeah.
Melatonin is highly sensitive to light, right?
Yeah.
Because it will degrade extremely fast, right? But you're showing basically that your bright light is actually producing a more amount of.
It's a night clot. It's night flight. So they had the time during the day, but the night they didn't have. The night was 0.2 knots. So it is in response what they had during the daytime. So yeah.
So it is basically the
It's
carrying forward.
carry over phenomenon. So what you sense during the day may have the consequence at night. So it's not that the machinery is not active, it's because the NAT is not there, N-acetyltransferase is not there, therefore there is no melatonin production. So let's see seasonal bird, because these birds are, think about this tiny bird, it goes 3,500 kilometers, come to us, takes another 3,500 kilometers at least, goes again back to its home, and if you analyze it, you see what it does. It does these two journey, and it is in flight, and it also makes this stopover. And if you translate it in terms of metabolomics and energy homeostasis, it's mind-boggling how this tiny bird maintains. And this was my interest to understand that how this bird goes into these cycles, and what are the molecular mechanisms into it. Now if you see this circle, this bird can't afford, okay, I become lazy, I will not acquire new feathers, because if it does not acquire new feather in sufficient time, it will not initiate the migration, so it will not come to India, for instance, and therefore on the way it might die. Same way, there is a lot of competition for the breeding site. They are wild animals, they have to find their territory. The male has to find their, make the house, because females arrive late. So the male's responsibility to defend the territory, so they acquire the territory, fight over it, then they make the nest and then wait for female to come, and they quickly, within 2 months, 8 weeks, they have to do everything if they want to have the fledglings. So 21 days incubation, 21 days is fledgling, and then 42 days, and basically they don't have more than 3 months good time, good season there anyway. So now in 3 months they have to organize everything. So it's a lot of biology, lot of processes happening. And if one understands, one can build up that what is the rate at which the systems function, a biological system functions. Because ultimately what we work only for how to make our life a little bit better. So these are the two species in which the lab works. Both are come to India and go back, and what we do, we nicely in captivity induce the way we want in different and then we record their rhythms. So we put them into cages. We provide, this is a computer generated system, data acquisition system. It plots, it does everything. When I started, I used to count just by eyes. Then I used to have a line and decibel recorder, pasting chart. But now everything is computerized, so the life is much easier than it was. Yeah.
This brings me to your question, basically, your earlier slide. It was actually, if you go to your earlier slide, it is actually moving from one particular country to another country.
Yeah.
For us, if you move, basically we actually get a jet lag or something,
Yeah.
or face
Yeah.
response, everything completely changes.
Yeah.
Now, the bird is actually having the same circadian rhythms. It moves from one country to another country, and when you're actually recording the actogram within a short time or something like that, do you see any complete out of phases in your actograms or something?
Yeah. So you can create that paradigm, jet lag paradigm. You would get that. So we publish in House Star and others. Now coming to the second question.
About the actogram, how it actually.
Yeah. So how faithfully it reflects what it did in nature. So this is latitudinal migrant.
Okay.
So it goes only across the latitude. It's not a longitudinal migrant. So there are longitudinal migrants, but this is only latitudinal migrant. So obviously we have to create a jet lag system. It travels not in single night, because this is a small bird. So it flies. So if you see here, it flies at night, then take a rest here, And this is this is a mass activity because it's in experiment situation. So it virtually forages here and then gets ready for the next night. That's what it does. So it is resting also. And if it flies, let's say 300 or 500 kilometers in a day, along the only a little bit direct change in the longitude otherwise, so it doesn't get too much jet lag.
Too much phase
Yeah,
difference.
no, so timing is not much.
Too much.
Yeah, it's just only the duration of the tail length is different, because at higher latitude they have the wider change in the huge variation in the
Did
tail
you
length.
ever undergoes any forced desynchronization or anything?
No, we haven't done any because this model doesn't go. So forced desynchrony protocol we don't utilize, but I have done for other regions lots of this T-cycle paradigm. Those data we have published, and currently we are doing some data. So what is the flexibility? How much we can tear apart? So we go at as less as 7-hour cycles too. So those kind of thing, but we keep on doing. So this is the clock, which I'm sure Dr. Sriram understands much more, but just to give a reference, because we know all the genes, so therefore we can create those seasonal changes and measure these genes, and so that's what we did, and our prediction was very the same. The prediction was that there are two ways the internal clock is going to respond to the external environment. A, it might change its phase, so different molecules which constitute the clock, they show change in phase, or they show change in the amplitude. So this is the two predictions which we set out at the beginning, and then we did also, this was the experiment, we created different stages, with phenology, we confirmed them, and then with PCR we measured it. So, and then if you see all these important genes, they were rhythmic in all the phases. That's what we expected, not only in one tissue, in a lot of tissues. We did all them. The paper has come out in FASEB in 2015, and then we said, okay, take the example of one just to explain. Let's say take a BMAL as one example. So this is all in 4 different times, so 4 different stages, and this is in liver, this is in the hypothalamus, and if you see here, so there is a change in the phase as well as change in the amplitude. So the way we think that the clock adjusts to the external environment by changing in the phase and changing in the amplitude. Now we then thought of that, okay, let's extend this story a little further, and we got interested in doing the RNA-seq. Subhajit joined lab a year ago. He's the only bioinformatician, only person who knows all this. I know nothing about it, but I know nothing about whatever I do anyway, except the theory. So so we found so this paper is from liver, and we have just submitted day before yesterday only. And let's see what molecular ecology does, but we submitted anyway. So these many genes we could annotate, and with the flycatcher genome, because this is de novo, we don't have the genome sequence, and these many genes in one phase were upregulated, these many genes in the other phase were regulated, some were common, and there were total all these genes were cycling. So there are a lot of cycling genes which we, so the next we have because 6 animals we are in each group, so we have plotted them out, but if you plot them, you make a heat map in terms of the timing. It is 1 hour after lights on or 5 hour lights on. This is at night, so if you relatively see here, most of the things happening at GT17 here relatively, and this GT17 here. Now we have only n is equal to 1 at each time point because of the cost factor involved and so on and so forth. But this is a first data in the literature showing that something is changing even at the transcript level. Now the students are further, so the next thing what you do based on you annotate and then relate and say we have so many pathways. and we still don't understand what these are doing, but we're trying to make some sense, and then we are carrying out further the work, and then we have other set of data in day-night changes, and goes on, and this is the ROS from ROS 454, that was Illumina. This is ROS 454, just to have bigger, because when I started 2009. Well, nobody was starting, but then I didn't know how to do it. People took the money and never did it. So that's what happens when you're a biologist, foray into your area. But that's what the science keeps you happy, at least me happy. And these are the some, they happen only one thing. Now, just 5 minutes more. We'll give it 40 minutes, 35 minutes, so 5 minutes more. So these, you see, this is a cartoon. This perfect, right? If mother or father feeds the baby insects, and it never goes out. And we all know, even light goes out, and if you are having something in your plate, it always goes into mouth. It never goes in nose or eyes. For some reason, it's just perfect synchrony. So we don't notice about the clocks. That's what the people didn't notice about the clock a long time ago. It is still people, unless Nobel Prize came, we used to talk, because this was not the first year they were nominated. For last many years they were nominated, and actually the field would have got in 90s with Dr. Asaf and Dr. Colin Keating. They deserved much more than anybody else, but the field came only in 65. So and practicality of the field was not shown at that time. So that was the reason. So now we think that what goes wrong? If anything goes wrong, things are going wrong. And the lab has some, interest into this. In all for this, we have just started this one on humans. So a student is now, running here and there. It's very difficult to get the human data, and most of us lie when we fill out the questionnaire. So that is, and one good example is our own students, because I have 25 chronobiology students every year, and I get them to fill the same questionnaire, change orders 4 times in semester. So that's how we generated 100. And the same students fills every time different, and error is about 40%. So they think that they are very smart. They don't fill out at the same time. After 7 days, when they have to give the diary, they say, Monday को तो मैं इस वक्त सोया था या जगा था, अपने आप भर देते हैं. तू कितने टाइम पे लिखे तो मैं कितने टाइम पे लिखा, likewise. But anyway, but that is what the beauty of working these innocent animals. So that's what we did. We started work on crows. And this is a learning paradigm, very simple paradigm. They learn whether the food, the space of the food, or the food is provided only at one place, and they made to learn first this, or the food is associated with one of these pattern of the cards, and we change the place, and these crows do beautifully. They learn it. So we after they have learned it, we test them after 1 hour and after 24 hours if they have retained it, memory retention, and expose them into, you see, this is the, the time tested protocol for humans. So if they are tortured, that's what they are kept into, they are never allowed to sleep here, or at least sporadic sleeping, and here they have the consolidated sleeping. Remember, this is one evolution which has happened, the consolidation of sleep. So it is very important sleep, and my colleague Russell Foster, who is Oxford, we came from the same mentor, and he has got 5 million pounds to teach British kids that you sleep more and sleep better. 5 million pounds grant in Oxford Medical School, and because the kids are not sleeping, and because I made it a technical talk, otherwise I have another talk where I saw the Twitter result and these results, and card result and so on. And now girls are worse than the boys, because they check more times their cell phone at night than the boys do, and so on, unless the boy has become girl. So it's a behavioral attitude. Now, so crows, you put them there, then you see the interesting result, right? So they are in continuous light, their activity is disorganized. We take this behavioral essay to show that there is disruption in their timing and circadian organization, and look, regardless whatever pattern, whether it is spatial pattern, they take longer time, they make more errors if they have been in constant light. That means they didn't have they sleep well, they would not sleep, and they also, it doesn't matter which protocol you use, and they do it. When we did it, people said, we published the paper in behavioral brain research, and I presented somewhere, and we are cynical people, so peer group is always cynical, they should be, and they said, where is the constant and light available? Fair enough. Let's do the light at night available. And I had a long time, because these are friends who run this light at night program in Europe. They have multi-billion euro project at light at night. So I said, okay. So I asked my student, go around Delhi University campus and find out where the crows potentially live, what is the light. So in the hostel, near hostel, roads and wherever. And we found anything from 2 to 20 lux light available. So we took an average and we came up with 6 lux. So I said, as the first thing, you do the 6 lux light. So day, night, 150 lux during the day, because anything above 100 lux is okay for us. This light is not more than 70 lux, which seems okay for us. So 100 lux is comfortable. Then we did this experiment and we replaced, we said, okay, we use and we measure all parameters. We said activity, we measured, sleep, and then nocturnal activity, so they were active, and the sleep rest duration was reduced as one would expect. And then we say, okay, we also did the behavioral essay, because I'm not a neurobiologist and we don't have also equipment. So, but if you don't have equipment, there are always alternatives to do it. And therefore, we said, okay, behavioral posture we will do. We did the behavioral posture and came out with this, that the sleep duration was really reduced duration was reduced. So although they were frequently sleeping, but the total duration, that's what happens if many of us put light at night. Never do this. I used to do it during my PhD days, because I didn't have watch my time. My research involved photoperiodium, so I had to turn on the light at 1 hour after. So I kept my room always lighted, and it took me a couple of years to come out of there. So I'm so paranoid that if I go in second AC in a train, I'm the one looking that if everybody sleeps, I go and turn on the light, because some people don't turn on the light, but I can't sleep. And I would advise all, unless you really sleep ventrally, so you're with belly on the bed, because then your eyes are there. Now, so if you see this mood, so this is interesting, because we are very should consider what the mood is there, because mood defines our relationships. So therefore, here grooming behavior and feeding behavior of crows, both were changed, and so much so that these crows started self-mutilation, which we defined pecking their own feathers. And then when we switched the protocol, the one which we are controlled, they started doing, the one which we are doing, they stopped doing. So that I removed all neurobiology slides, because that was not the point to show here. So this is one case how they pluck their feathers off, and these guys are not doing the same thing. They are from the same flock, and this is the score we have given. So this even in exploration of the object, which tells us about the cognitive ability. So this is also impaired. And nowadays you people must be reading lots of Times of India stories when foreigners are publishing. You don't say kids. My grandchild, they all love to watch cartoon, because their mothers, that keeps their mothers happy, because they don't have to do any extra effort, or fathers happy, because once they get the mobile, they are free, and that's what my grandson, bile. So immediately when the milk comes, he says, where is the bile? Bile. And he doesn't, he has started kind of, Baba, that means you go, otherwise you will object to the bile. But this bile is going to really be very nasty for them in coming times, because both, you measure the variable, it's changing, whether it's approach, whether it's a latency, and so on and so forth. So what really I tried in 38 to 40 minutes, I tried to use some data based on birds, that organisms have the sense of time. They measure it also, they translate it also, and they use it also to organize and regulate. Remember, clock does not regulate the activity, it does only organize the activity in time. So it regulates time. So animal will still be active, but will not be active at the right time. So that's what the clock does, and that probably, because we always look the profitable times are there. So I again thank Dr. Sriram for asking me to come here, and you people for being here. Thank you so much.
Any questions?
Yeah. Curiosity, there may not be question. If any question, otherwise.
Yeah, very interesting and deep talk. I was just wondering, there is one sense which I was told about, I'm not sure how true it is, that the need for sleep is such as like 8 hours sleep is a rule that is told. Apparently I was told that that's not really a rule, I mean, that you don't need it. In the context of humans, of course.
Yeah.
Do you think that's factually true in terms of?
Yeah. So the sleep is regulated by two processes. One is homeostatic state. That means the time you wake up in the morning, there is a, it builds up. So the more time you are awake, there is a more urge to sleep, right? But there, in science, there are two things, that the necessary and sufficient. And that what individual's body decides. So that what decides individual, but we all have individual requirement of sleep. That means individual requirement of growth and repair. Because sleep period is for repair. Because that is when growth hormone is secreted. It's not secret. It's very low, in amount during the daytime. The growth hormone which helps us to repair. So it all depends what we were doing, and it all whether it is sufficient for me, for instance. So for last 3 nights, today I slept like 6 hours, 7 hours, but the 2 nights before I slept only 4 or 5 hours, because all grandchildren were there in the house. So I had to quit my bed, and then they would sleep during the daytime, keep running up to one, because their birthday was there. So you can't say also, right? So they all except me. So I was sleep debt. I had a lot of sleep debt, and it still have. So my cognitive ability, for instance, is not really up to the mark. The one way is, so then there are other factors like food, what food you are eating. So if you don't eat or drink only water or very small amount, probably you can. And now we have the drugs which can keep us for 72 hours awake. So that's what US Army does. That's what many scientists does. The Kalinga Awardi, who is a friend, Olympic, he came to our lab, and he didn't take, he took the pill, which is very close relative of Viagra. The same chemistry, little different only. It makes you 72 hours awake. So that's not true that 8 hours is there. What is required? The cycle, each cycle is 90 minutes. So composed of non-REM and REM. So as the time goes by, in first cycle REM component is small, non-REM is very large, and then the second cycle REM becomes, so it's 3 hours, right? Then 4.5 hours. Sometimes after 4.5 hours you woke up during the REM and you feel very fresh, because you think you don't require it. But believe me, next day you are going to feel tired. So individual requirement, but I would advise that you should sleep as long as you require really. Don't wake up forcefully. Use alarm clock as little as possible. That's the thumb rule. So if you sleep, many people confuse circadian clock regulation with sleep apnea. Sleep apnea is a kind of disease. So where that has nothing to do. But if you sleep on time, that's good. If you cannot, you delay it. But you delay it, you don't frequently change it. You delay it. We can always delay it. So we can live on, let's say, Germany time or British time, or we decide to we live on San Francisco time. If we can afford to, if our house is dark enough. That's what many people do. So my son comes every day around, because he comes from Gurgaon every day. So 12 o'clock, 11 o'clock is good time for him, 11 to 12. And then the only thing I have, kind of advised my daughter-in-law that let him sleep, because he has very flickery sleep. He's a computer wizard. And this is very dangerous with software students, because software students don't realize, because there is not a physical work involved. So they don't get otherwise tired. Muscles don't get tired, their brain gets tired. So they think that they are not tired. So they can't sleep so easily. And then if they have to formulate an algorithm, and if they are working, because company pays them to do this, so they keep on thinking. Then if you ask them to sleep, so they turn off the light, but it's still working on the computer. And then so much so it becomes ingredient. Students in IITs and university institutions, if they are doing at night, they think that they are genius. And this is the anti-biology, but that's what people think. So I think regardless of what people say, each and every one of us doesn't have. Some people question me, our Prime Minister sleeps only 5 hours. So I ask him, are you a Prime Minister? Is your food is also tested by 27 people before it comes to you? Have you doctors who know that what your body requires? These people, late Dr. Abul Kalam, APJ Kalam, he did not know that he had diabetes. This was told by his physician, who is the chairman of Gangara, because he and I were the same committee. So he disclosed this. He said he did not know that he had diabetes unless he became the president. So when we were appointed, that was the first test we did, and we found out that he had the diabetes. So these people, famous story, if you remember, of Mr. Narasimha Rao, our Prime Minister. If before Prime Minister, if you have his Google photo, you go Google, and after it becomes, because we secrete a lot of dopamine. Think about the day for students, your sister is getting married or your brother is getting married. Do you feel sleepy? You don't. You pumped in. Or you are going to have a grant announcement in a positive, and you don't feel sleepy. So these are other, but as I said before, that please don't use alarm clock until or unless, because essential, because corticosteroid is there to tell you, this cortisol is going to tell you that please wake up. Never pick up a fight with a boy during the morning. If you want to pick up the fight, the best time to pick up the fight in the morning. जैसे उठे आप देखिए बच्चा सुबह उठता कहते अरे मम्मी प्लीज 5 मिनट और सोने दीजिए मम्मी प्लीज और आप डिस्टर्ब करिए उसको तो टेस्टोस्टेरोन इज हाई फिजिकल टॉलरेंस एंड कार्टिकोस्टेरोन एंड देन पिक अप द फाइट कभी नहीं शाम को आप फाइट करिए बोलेगा ही नहीं तो these are knowledge करते anyway yeah madam
I have a question. So these circadian rhythms, you mentioned the daily rhythms and the calendars. So I was curious, in absence of additional triggers like temperature or seasonal changes, can you, for example in birds, the migration instinct, if there are these external signals absent, do you still think that the circadian rhythms will work?
So migration is controlled by circannual clocks. So that's the time of migration. That's lab also. So I'm one kind of, so recently I wrote in encyclopedia the article. So there is a long-term rhythm, circannual rhythms. But the daily alteration is done by circadian clocks. So this is the time to migrate at night and not to migrate. So not to undertake journey or stuff. This is under circadian control. But when to migrate in the year, that is a circadian clock. And now the last 4 years we have pumped up like 10 papers in the literature that these circadian clocks show seasonal alteration in phase and amplitude. So this is our hypothesis is that this is the way circadian clock connects to seasonal changes, which is otherwise regulated by circadian clock. Now, we don't know where the circadian clock is located. So that's the problem. So we are trying, we have some good hypothesis, but we are not near to that finding right now.
Any other questions? Okay, if there are no other questions, let us thank Dr. Vinod Kumar for taking his time out to come and give a very wonderful and very exciting talk about the circadian rhythms and also about the circadian rhythm. Incidentally, I forgot to tell you, basically, he mentioned it basically, this year's Nobel Prize has gone to chronobiology, and it has been given to, awarded to Jeffrey Hall, Michael Rosbash, and who is the other guy? Michael Rosbash.
Michael Young.
Yeah, Michael Young. And Rosbash was
Not
actually
so young
he was working in his lab actually. So it is really a privilege to.
Yeah, so SERB who funded, so they needed to show there is a, nowadays our Prime Minister asked everybody to show what are you doing with the money. The Science and Engineering Research Board, so they also needed to put, this was in Chennai only this month, India International Exhibition something. So every department has to, so science department also. So I received a phone call that can you send me a poster. I said I'm traveling but I'll arrange. So I asked my former students who will, so I have 4 labs, Lucknow, Delhi, Meerut and Nehu. So SERB school in chronobiology connects to Nobel 2017. Incidentally that was very hit. And that was the only thing DST had to show. Other was microgravity. So when the 2 things came, I was very kind of pleased that microgravity is something which is really very useful. But if the chronobiology now is being shown just next to microgravity and attracting, so really it's good. But I always request people that please you don't leave whatever you're doing. Only start thinking if that makes anything in terms of time. That's what you need to do.
Yeah, one final thing is that I didn't know that till he told, like he is also a student of Professor M.K. Chandrasekharan. He is one of the pioneer in actually circadian biology. He has trained host of students actually. I have read a lot of his papers from the experimental point of view. It was really an exciting paper. I think he worked on cockroaches and
later on bats.
Yes.
Mice, Drosophila.
Yes, and so
Unfortunately now.
really it was our privilege to have you here. And as a token of our appreciation, we would like to give this to you.
Thank you, sir.
So once again.
So please go ahead and for this.
Yeah.
Please visit whenever you are coming to us.
So,